How's everyone today? Is all doing okay?
Yeah.
All right, good. Fantastic. So yeah, we're gonna just jump in and get as far as we can with this. We spent a lot of time last time laying down the biology review, right, showing how cells do what they do, and for good reason, right? So we learned about how cells replicate, how cells synthesize proteins for the body. We learned about sensitivity of cells to radiation. We learned about some rules for cell sensitivity to radiation, right? Recall the law of Bergoni and Trabando. So we're going to sort of pick up where we left off on that today and keep on going. And we finished the last thing we talked about last time, right? Were these, let me get to the slide here, were these radiation response curves, right? These graphs that plot the response of the body or a population to exposure to radiation, right? We talked about linear graphs, nonlinear graphs, graphs with a threshold, graphs without a threshold. And so we're going to sort of continue to elaborate on some of that stuff today. I want to start with this idea. Talking about cell damage, that's where we're going to get into is how cells get damaged, how this damage manifests, and what are the consequences of it. So we're going to start off with this idea called target theory. Okay, so this is chapter 41 if you wanted to follow along. Target theory just says very simply, right, that there are certain molecules in the cell that are necessary for the cell to survive, right, critical to the survival of the cell. We call these molecules target molecules. Okay, what might be an example of a target molecule? Something that's critical for the cell's survival.
DNA.
Yeah, the big obvious one's DNA, right? It's the largest molecule. It's the largest large molecule in the body, right? It's this huge, long thing. It is millimeters or so in length. If you actually stretched out one chromosome all the way stretched out, it would be macroscopic in length, okay? Interestingly, if you took side note, if you took all of the DNA in a single cell and stretched it out, it would be longer than you. Okay, just one of your cells, all of the DNA would be over 2 meters long. Right? If you took all 23 pairs of chromosomes and stretched them all out, right? So DNA is a huge molecule, right? It's a microscopic thing, but it really is a very huge molecule. So it's very sort of a trick of nature how we've been able to package so much information to this tiny area. Anyway, so DNA is a target molecule. But DNA doesn't leave the nucleus, right? So a cell that wants to use DNA to build things, how does the information get out of the nucleus? Some other nucleic acid has to do it, right? What takes the information out of the nucleus of the cell?
Is it a fluid?
Not a fluid. I mean, the cytoplasm is what everything's sort of floating around in, right? But there's ribosomes help, right? They're a helper protein. I'm looking for RNA, right? Specifically, mRNA, right? Messenger ribonucleic acid. So let me just briefly go back. I know it's been an entire week since we talked. And remember that the way proteins are built for the cell is that the cell makes this strand, this mirrored copy of a DNA strand, and they call it mRNA, or messenger ribonucleic acid. Okay? This mRNA strand is a mirrored copy of some section of the DNA in the nucleus, right? And that's brought out of the nucleus. Okay? We use that mirrored copy. Remember, I used the analogy of copying a book in a mirror, right? So this is the mirrored copy of a book in a library. Took it out of the library, and we're going to use this mirrored copy to build a protein. Okay, ribosomes transfer RNA attached to amino acids, do the job of like building the protein, but the instructions, right, the manual for building the protein is the messenger RNA strand. Okay, so this nucleic acid, if it gets damaged, that's critical to the cell's survival. So mRNA counts. Okay, as well. tRNA, transfer ribonucleic acid, the one that does the job of lining up the amino acids in a specific order, the amino acids that will eventually become a protein. These guys, they have tRNA. That tRNA, those tRNA molecules are target molecules. So when we think about target molecules, what we're really talking about are the nucleic acids, okay? How much of a cell's structure is nucleic acid? Like by percentage? 100%? 50%? 10%? So it was the first thing I told you guys last class. Last class. Here you go. The body is the body, meaning a cell, 80% water, 15% proteins, 2% lipids, 1% carbohydrates, 1% nucleic acids. 99 parts out of 100 in a cell are not nucleic acids, right? Only 1% of things in a cell count as nucleic acids. And those are the target molecules, okay? So when we say there's a theory called target theory, where we call some molecules target molecules, and if they are hit, those are critical to the cell's survival, and those can cause irreversible cell death, damage, mutation, right? If any non-target molecule is hit, and again, what we mean by hit is just a particle strikes it. Alpha, beta, gamma, or X-ray photon strikes it, right? That damage is sublethal. So target molecules strike to a target molecule, likely lethal to the cell's survival. Non-target molecules, basically everything else in the cell, sublethal. Now, sublethal can still hurt you, right? A taser is sublethal, and if I tase you, you would not be very happy, right? But you wouldn't be dead, yeah? So there are sublethal things that can happen that are still not good, right? So when we say sublethal, we don't mean not harmful. We just mean not going to cause death, okay? But you can cause other things to happen. You know, there's other mutations that may happen. But anyways, what we call a hit, in quotations, is defined as unrepaired functional damage to one of these target molecules, in this case chromosome, and which may result in deactivation of a portion of the genetic code. If a portion of the genetic code that would be used is deactivated, what happens?
It's killed.
What do you mean? How do you mean, Joe?
I'm reading along with the
Yeah.
chapter, and it just describes as it kills bacterium, the target molecule.
Right, so like a bacterium wouldn't survive, right? Because the genetic code gives us the instructions to build proteins, right? And proteins are necessary for the cell's function. So if you destroy the instructions, then the cell can't build the parts that it needs, and then will die, right? So lethal to the cell's survival. Importantly, these hits are not just a single event. Like we sort of have this cartoon picture, and I mean, they literally even give you the cartoon picture, right, of a single photon hitting a single strand of DNA, right? But it's important to know that this is like a sum of events, right? There will be many, many of these ionizing events in and around the target area, and it's this accumulation of these ionizing events that we call a hit. So it's not just a single photon and a single ionization. This can be many, many of them making this happen. Okay, so that's the idea, right? We have DNA. Hits are strikes to nucleic acids, DNA, RNA, and they can either be direct strikes or indirect. A direct hit is exactly that. A photon is on the right path, and a piece of nucleic acid is in the way, and the photon quite literally strikes it, ionizing some of the molecules, right? Ionizing some of the atoms in the molecule and breaking the molecule apart. Okay. An indirect hit, which is way more likely, is a strike of water, right? Water in the cytoplasm accounts for about 3 parts out of 4, roughly 75-80% of a cell, 8 parts in 10. And that's so much more likely to happen, is that the water molecules nearby to the nucleic acids are struck. Those water molecules are, for one, there's more of them, and 2, they're easier to ionize. And when you do ionize these water molecules, they become highly reactive. They tend to share or borrow electrons from nearby atoms, and that can tear apart DNA. That's what we're going to try to learn about here in these upcoming slides. So an indirect hit consists of the creation from water. That doesn't seem right. I'm going to read it, then we'll see if that sounds right. The creation from water of chemical compounds, which then attack the target molecule. Basically, you have water. If you ionize water, it turns into other reactive substances, which we'll discuss, and those reactive substances can then act on the nearby DNA molecules. It's much more likely to ionize water than it is to directly strike a molecule of DNA. This idea of an indirect hit, we refer to it as radiolysis of water. Radiolysis. Help us with this word. What do you see in this word that's useful? Radio tells you what?
Radiation.
Tells you it's dealing with radiation, right? What is lys? L-Y-S.
Complete or through? Like dialysis, right?
Dia is the complete or through part of it. Lysis is breakdown. Good thoughts, because you got the right word, right? So lys, for example, in your cells you have little enzymes called I almost lost it. Lysosomes, right? They are enzymes that break down worn out, used up chemicals for repurposing. Lys means to break down something. So radiolysis of water says radiation is breaking down the water molecules. Can also be called hydrolysis or hydrolysis. Ionization causes the chemical breaking of water into various things, ions and free radicals. So let's define what an ion is, what a free radical is, and see how those affect things. So again, this is dealing with strikes of the water molecules in our cells, which is the most likely interaction with radiation because as you learn, most of a cell is water, most of us is water, so interaction with water is the most likely thing. An ion is the term we give to any charged particle. So an atom that has a net charge, that's an ion. A single electron with a net charge, or any atomic particle that has a net positive or negative charge, a charged particle, that's an ion. It's where we get the term ionization from, right? The creation of a charged particle. But an ion is just that, a charged particle. Occasionally you'll hear us refer to something as something called an ion pair. That's an atom which is broken apart, and you have two charged particles, a pair of charged particles that came from a single atom. So that's what an ion is. A free radical is slightly different. Free radicals are uncharged atoms. So no electric charge, they're neutrally charged. Otherwise, you would think a free radical would be a happy atom, okay? But the best example is hydrogen. Hydrogen is uncharged, neutral, and so all hydrogen is, right? All hydrogen is a positively charged nucleus and a single electron orbiting around the only shell of hydrogen, okay? One proton, one electron, okay? This is a little hydrogen atom, simplest atom, but hydrogen is considered a free radical, okay? When it's on its own, not attached to anything. What is hydrogen usually attached to inside of our cells or inside of our body? To make water, right? H2O, two hydrogens, one oxygen molecule combined to make water, right? So usually hydrogen is floating around attached to oxygen molecules and it's paired up and happy, okay? But occasionally when there's an ionization event, for example, when water gets ionized, those little hydrogen molecules can break off of the water molecule, okay? And when they do, you have a free hydrogen molecule floating around freely. We call it a free radical, okay? Now it's uncharged, so there's nothing like electrically that's going to attract or repel this atom from any other atoms nearby. However, this atom only has one electron in its outer shell. And the first shell of an atom, this innermost shell, or if it's the only shell like hydrogen, how many electrons fit into that shell? You're thinking the right thing, but for bigger atoms. A hydrogen atom with only one shell, how many electrons can the innermost shell of an atom hold? Two, right? So if you're hydrogen and you only have one shell, and that only shell only has one electron, even though the atom is uncharged, it is going to try to rob an electron from a nearby atom, okay, pulling an electron from a nearby atom so that it can fill its outer shell, okay? Even though it has no electric charge, it will still seek out a chemical reaction with nearby atoms, okay? And that's a problem, right? this little free radical is going to reach out and rip electrons off of maybe a DNA molecule. And that's going to change the way the DNA molecule is connected to its other parts, okay? So this free radical can cause harm the same way an ionized particle can cause harm, okay? The way you get free radicals is by ionizing water, by breaking apart water, okay? This is one of the outcomes of breaking apart water, free radicals.
So this is hydrogen peroxide.
Not yet, but yeah, you're getting there. We're gonna get there.
That's that process.
Yeah,
that's why it works to kill germs.
Right, because hydrogen peroxide, H2O2, will try to steal.
It'll steal from the living germs.
Yeah, it's gonna
That's
rob
why the
electrons
germs
and
die.
exactly rips them apart.
Okay.
Yeah, breaks them down.
I'm just looking for a
Yeah.
solution.
No, you're not. No, you're not. Okay, yeah, you're on the right track. Exactly. So that's the idea, right? We have water in our body. When water, which is easy to ionize, when water does get ionized, we make two types of things, free radicals and ions. Both of these are chemically damaging to any nearby molecules. Maybe your DNA is nearby. Okay, and again, the best and really the only example you have to remember is hydrogen. Okay, hydrogen is a free radical. All right, good. So that's the idea. We're going to rob nearby electrons from an atom, from another atom, and we're going to make chemical changes to nearby molecules because of that. Okay, good. One of the processes that happens here is shown here. Okay, zoom in on this a little bit. So from left to right, the process is being shown. You're seeing the picture of the molecules, and then down below, you're seeing the written out explanation of what these molecules are doing. So two water molecules, which there will be lots of water molecules right next to each other. So two water molecules just hanging out, minding their own business on the left side of the screen. And you see the right side water molecule, this one, is struck by an X-ray photon, okay? That strike that has enough energy, may have enough energy to charge the particle, to make the water molecule have a greater charge than it used to. That's ionization, okay? So you'll end up with two charged particles, okay? You get one that has a net positive charge and one that has a net negative charge. And it's just because if you charge one molecule, the one nearby will have a lesser charge. There will be a difference in charge between the two. Even though one of them wasn't charged at all, because you charged one of them, now you'll have two that have a difference in charge. It's just because you had charged one of them. So you have HOH plus and HOH minus. Those two can then split. So you see HOH, hydrogen, oxygen, hydrogen, HOH. Negative, negatively charged, HOH positive, positively charged. These two can further split into four different molecules. And so this is the breakdown process of water. You ionize a single water molecule and it makes this cascade of events to get you to here, where you have now four molecules, two of which have a net charge and two of which are considered free radicals, okay? So we see a net negative charge in this oxygen-hydrogen pair. We see no charge in this hydrogen atom. This is a free radical. We see no charge in this hydrogen-oxygen pair, free radical. And we see a net positive charge in this hydrogen atom. So this process of ionizing a single water molecule, the outcome can be four molecules that all four of them can now cause harm.
So the free radicals, they're unpaired electrons, correct?
They are atoms with unpaired electrons in their outer shell. So, hydrogen is really the best example, right? You have a single electron, that atom's going to seek to try to get an extra one. So now you've got, but the moral of the story is, which we're going to try to get to, the real moral, is that you have four charged particles that can be created from just two water molecules nearby, one of them getting charged, one of them getting ionized, okay? And all four of these are now going to be able to cause some harm. So you have free radicals and ions created from that pair. So this can now further chemical complication. Oxygen can combine with a hydrogen radical to form hydroperoxyl radicals. So here you have O2, which you have plenty of O2 just floating around, right? You have oxygen in the cells of your body, yeah, and in the bloodstream, all that kind of stuff. So you have an oxygen molecule that just is trying to mind its own business, okay? And one of those free radicals of hydrogen can happen to be nearby to that oxygen molecule, okay? So this is, let's zoom in A. Here's your hydrogen free radical that was one of the outcomes, one of the products of ionizing water, okay? The other three particles we're not talking about right now. So this is floating around, free radical, ready to form a bond with anything nearby. And O2, two oxygen molecules happen to be nearby, okay? And it just so happens that when these two combine, they create HO2, two oxygen, O2, and a single hydrogen. This is now a free radical again, okay? I won't go through the chemical makeup of this, how many protons, how many electrons, how many unpaired, but take my word for it, this is a free radical. We still have an unpaired electron in this new molecule that's been created. So there's a free radical ready to go do some harm. That free radical, the one that was up there, is now being shown here. Well, maybe there was another one nearby, because there's many of these events, right? So another one of these, two of these molecules, two HO2 molecules, can combine to form H2O2 plus an O2 molecule, okay? This is not harmful, it's just oxygen, but that's harmful. H2O2 is hydrogen peroxide, okay? There's a hydrogen molecule per oxygen molecule, hydrogen peroxide, okay? So there you go. We just I gave a hand wavy explanation, but an explanation nonetheless of how you can get from water molecules combined with some oxygen molecules and it is cascading process to get you to H2O2 from just regular water, okay? It wouldn't do that on its own, but it can do that with an ionization event. Okay, and many of these ionization events can make many more hydrogen peroxide molecules. And you know hydrogen peroxide is harmful to living things because we use it as an antiseptic, right? We use it to destroy living organisms on surfaces, even on, it's not great to use on cuts because of this exact reason, but some people use it on open cuts and things like that to, sanitize, disinfect wounds. But yeah, this is also parenthetically why doctors do not recommend putting hydrogen peroxide on a cut or something like that, because it damages the cells at that area. Okay. Yeah, that's the whole story here, is that hydrogen peroxide, which you can make from water, is harmful to living things. Okay. All of the chemical products created from radiolysis of water can chemically attack target molecules. So all of the ions, free radicals, hydrogen peroxide that can be produced from ionizing water, all of that can create indirect hits and damage these target molecules. Direct hits, yeah, go ahead.
So would that fall over the runge damage?
One more time, the rung damage?
Yes.
So rung damage will be from an indirect or direct hit.
Okay.
And this is what I was about to say, it's so much more likely that damage to target molecules, DNA, RNA, that damage to target molecules will be from an indirect hit, much more likely than it would be from a direct strike. And we've just given the explanation of why. You have way more water molecules in your body than you do target molecules, and these water molecules are, one, easy to ionize, and when you do ionize them, they form free radicals and ions, which can then seek out and damage those target molecules. Okay, so direct hits are low probability compared to indirect hits. It's more likely to interact with water than it is to interact directly with the DNA. So that's one problem. These are functional damage to the chromosomes or any nucleic acids. Those are hits. That's target theory. There's another problem with things that can happen to cells, okay? And this is damage to the cell's membrane. What does the cell membrane do?
Keeps organism.
So I heard protects the organism. Sergio says
It has everything inside.
keeps the inside in, right? The outside out, right? Taylor's similar thing, right? Okay, so the membrane serves as a separator between outside and inside, right? Sort of like your skin keeps the outside out and the inside in, right? If you get a cut in your skin, you now have an opening for bacteria, viruses, stuff to get inside your body, right? It's no different for an individual cell. If you make a break in the cell's membrane, remember from science class we called cell membranes selectively permeable barriers, right? Things can get in and out, but only certain things, right? If you let just any old thing in or out, then a cell can be very, very hurt from that. For example, all of the cytoplasm can leave the cell, causing the cell to dry out. Okay. Now that can happen under certain circumstances, under like hypersalinity circumstances, yeah, where you have lots, or hyposalinity, low salt solution situations. So you can damage cells, removing the water from them just by osmosis. But this is a physical, literal break in the cell's membrane from radiation exposure, from ionization of the cell membrane. The layer around the cell membrane, is called a phospholipid bilayer. It is just a molecule connected up, one molecule connected in a big strand, bunch of these little molecules, and it forms a separator. Okay, that barrier can be broken. Toxins can enter the cell, the cytoplasm can dry out. Cell membrane damage is lethal to the cell. So that's one type of thing that can happen. This is not considered target theory. Remember, target theory is a strike, either direct or indirect, to the chromosomes or any nucleic acid. This is just another thing that can happen to the cells. So then we'll go now to let me backtrack. Okay, so we're going to go and we're going to talk about these chromosomal damages, right? So we call them hits. We're going to describe the different types of hits that can happen. But first, let's just break down what are the ways cells can die from radiation exposure. We've given the target theory and the theory of cell membrane damage. So let's see how those can manifest. In extremely high doses, the cell can die instantly. So instant cell death is one option. High doses of radiation would rupture the cell membrane, causing proteins to coagulate, the cell to dry out. This is instant cell death, absolutely lethal. This will occur in exposures upward of 1,000 gray. What's the unit, the gray? What is it for? What do we use that unit to talk about? To measure radiation, right? But we can measure radiation in different ways, right? We can measure exposure, we can measure absorbed dose, we can measure dose equivalent or effective dose. There's 4 different ways of measuring, right? So what is the gray the unit for? Exposure, absorbed dose, dose equivalent, or effective dose? Exposure and absorbed dose. Okay, we use the unit, the gray, to talk about exposure and absorbed dose. If you give a human 1 gray of radiation exposure to their whole body all at once, that could kill them. Okay, it wouldn't definitely. No one will die from less than 1 gray of exposure to the whole body. Somebody could die above a gray of exposure to the whole body. That is the threshold for human death, 1 gray. Okay, so 1,000 gray, 1,000 times more, right? Obviously lethal, right? 6 to 7 gray to the whole body would kill anybody. Okay, that not instant, but very imminent death to anyone from 6 to 7 gray. You do not survive that. So 1,000 gray, all of your cells destroyed immediately. You have basically, seconds, minutes to live. Okay. In an individual cell, it's the same way. An individual cell, instant cell death from 1,000 gray or more. Interphase death. Okay. What's interphase?
Doses between 1 gray and several hundred gray.
That's how much it takes to cause death during interphase. But what in the world is interphase? It's a part in the cell's life cycle, right? Remember the pie chart of the cell life cycle? So what do cells do? Go ahead, Abby, what do you got?
Divide. So interphase is part of it coming together and getting all the chromosomes to go
So let me bring up the slide here for it. It'll be, I think, maybe helpful. So here is your cell life cycle, right? Notice the sliver at the top, mitosis. That's cell division, okay? The rest of that pie chart, 90% of that pie chart is called interphase, okay? So this whole pie, all of this, except for that sliver at the top, mitosis, is called interphase, okay? Cells die during interphase because of a failure to produce the necessary proteins, okay? Gap 1, synthesis, and gap 2 are the three parts to interphase. Synthesis is where the cell is building all of it's replicating all of its DNA. Gap 1 and 2, we have protein synthesis happening. But importantly, if enough radiation exposure happens to the cell after mitosis, the cell can die during interphase because it can't build the parts that it needs. Okay, it can't build its components, so it can't function. So let me go back to here. Interphase death. The cell necrotizes and dies. Necrotizes is also a way to say dies. So it dies and dies within a few days after exposure. Remember, the average cell lifespan is 24 hours, so if we're talking about on average, we're talking about hours after exposure. The relationship between how quickly the cell dies and related to how much exposure is inverse. The more exposure a cell gets, the shorter its lifetime is. That's an inverse relationship, right? Cells die quicker with more exposure. That's the idea. So 1 gray, the cell will die. Several hundred gray, the cell will die quicker. And they say they are very correct to say the biochemical causes are obscure. Why? Because you can't see chromosomes during interphase. So if a cell dies during interphase and then they take that cell and put it under a microscope, they can't see the chromosomes to determine exactly what happened. Okay, we just can't see them. The molecules are too small for even our best microscopes, which means that we don't know exactly what's happening. We just know that death happens, okay, during interphase from exposure. We have theories, ideas, right, models of what happens, but no one can, absolutely nobody can observe those molecules dying because the molecules that we're talking about are smaller than the wavelength of visible light. And that means we can't observe them. So the biochemical causes are obscure, but we still have a pretty good idea about what's going on, nevertheless. Scientists are smart about things. If they can't directly see it, there are ways of indirectly seeing things. Reproductive cell death. The cell permanently loses its ability to procreate. At the next cell division, the daughter cells will die. So the cell gets to live out its life, but the daughter cells don't survive. This would be a problem during synthesis phase, where we're making the copy of the DNA. Right? So like you have all the parts that you need to work, but when you go to replicate, it doesn't build two functional cells now. So cell death at replication, mitosis. After mitosis, because the next one is mitotic cell death. Mitotic cell death, genetic mutations cause the daughter cells to die after the exposed cell has divided. So surviving offspring, then die quickly, okay? Instead of offspring never live to begin with. So to recap that, you have instant death from very, very high levels of exposure. You have interphase death from moderate levels of exposure. And then you have reproductive or mitotic death from medium to lower levels of exposure. Mitotic death can occur from small doses of radiation. Here's a word you're going to need to get very familiar with and make friends with this word. The word is stochastic. Let me circle it with a pen. I tried to highlight it, but you can't highlight a yellow word. Okay, at least in yellow you can't do it. So there you go. This word stochastic. When we say something is stochastic in nature, what we're saying is it's random. Take the word stochastic and in your head plug in the word random. That's literally what the word means, okay? So if something was non-stochastic in nature, what would that mean?
Random.
not random, which means you know what's going to happen, right? Deterministic is what we will call it, okay? So there are two types of effects of radiation. This is what we're going to be talking about coming up here. Two ways radiation can affect the body. Stochastically, we don't know what's going to happen, okay? Stochastic effects come from very low doses of radiation. Like, we give you an x-ray today, and then you get cancer 30 years later. Maybe it was caused from the x-ray exposure 30 years ago, but probably it was from something else, right? It's random in nature, okay? More x-ray exposure makes you at higher risk, right? So it's all about probabilities and randomness when you talk about stochastic effects. Deterministic effects of radiation, on the other hand, have a threshold. We know when they happen. They're determined by dose, okay? Like, I know that if I put you under a certain amount of radiation exposure, 2 sieverts, for example, you're going to get a sunburn, okay? And we have a threshold. We know you're going to get a sunburn, and we know the sunburn will keep getting worse with increases in exposure. The effects are deterministic. So those are the two ways you think about effects of radiation: stochastic or random, deterministic or we know what's going to happen, okay? Mitotic cell death is stochastic. We don't know how much radiation can cause it. We know that any amount could. Higher amounts make it more likely. Whereas if you looked at these other three, right? Reproductive cell death, there's your threshold, right? 1 gray is your threshold for mitotic cell death, yeah? 1 gray is your threshold for interphase cell death. 1,000 gray is your threshold for instant cell death. We know the thresholds for these, so these effects are deterministic or non-stochastic. All right, so start to make friends with that word stochastic. It's a word we use all the time. All right, let's review briefly ways DNA could be damaged, because it is just super interesting, I think. We're not reviewing it because I think it's interesting, but I do think it's interesting. All right, so 2 types of DNA damage. You have main chain scission. Scission like scissors or cutting, right? A break in the rails of the molecules. And then there's B, rung damage. The rungs are your ladder that you climb, the part of the ladder that you would climb on, right? The rungs of a ladder. These are breaks in the nitrogenous base pairs that make the rungs. We're going to refer to something in a little bit called a frame shift mutation. These are changes in the actual sequence of the genetic code. Both of these things can cause that. Okay, breaks in the rails, so like the ladder breaks and then the ladder reconnects to some other broken part of a ladder, right? That changes the instructions. Rung damage, removal of a base pair, also changes the instructions. So we call these changes in the instructions a frame shift mutation. Any change in the genetic code is a mutation. Mutations happen. There is nothing that you can do to stop them from happening. Even if we stopped all of the random background radiation exposure, if we stopped all medical radiation exposure, we would still mutate, okay? DNA mutation is how we have evolved, right? It's how any animal evolves. If our DNA was perfect and made a perfect hand-me-down to all of our offspring, no animals could ever evolve, right? Any individual organism could, act in a way that makes it better in its environment, but that organism couldn't pass on any traits to its offspring if we couldn't have random mutation, okay? We need random mutation for evolution, so it comes along for the ride, right? It just so happens that, 9.999 out of 10 random mutations are bad, right? Cause harm. Some very small fraction of random mutations are beneficial for an organism, okay? And this is an important thing to learn. The mutations we cause from radiation exposure are no different than the types of mutations that happen to a cell already, okay? You are not going to make any cell mutations that haven't already been happening, okay? Radiation just increases the rate of these mutations. There are more mutations in a given time with radiation exposure than without radiation exposure. We're going to circle back around to that idea because it is an important thing. All right, so here you go. Here are a couple of options for frame shift mutations. You have the main chain scission, where the rail gets broken. Now, importantly, if it's just a single rail, a single side of the DNA strand broken, that does not tend to cause a frame shift mutation unless the other side also breaks, okay? There might be some random weird things that can happen, but generally speaking, this is not going to cause a change in the genetic code because only one part of the ladder is broken. Okay? Still connected on the other side. However, in the middle, shown with B, if you broke both of the rails of the ladder, both of the rails of this ladder become ionized, break from an either direct or indirect hit. Now you have two parts of the ladder that will float apart, separate, right? Be knocked separate, and those will now attach in random spots to random other broken pieces of ladder, okay, or the end of another DNA strand. And that's going to make actually noticeable changes to the chromosomes. You can see them under a microscope. You have to do it at the right time, right, when the cell's right about to divide again. But you can see these changes because the chromosomes change in shape when there's damage to them. So this is one frame shift mutation, main chain incision, and then you have rung damage here, where ionization event causes a piece of these nitrogenous base pairs to just be broken off. Okay, and now where a piece of instruction would have been, it's now not there. Okay, and that's going to make all sorts of weird problems. The cell will either build the wrong protein, not build the protein at all, build too big of a protein or too small of a protein. It's a number of different things that can happen, but the point is we've made a change to the code with rung damage. All right, so A is repairable. B and C are not repairable. Okay, some final notes on mutation before we try to move on a little bit from that. Mutations are unrepaired damage, right? So that first one where one side of the rail was broken, that's not a mutation because that damage can be repaired and the genetic code is not altered. So mutations are unrepaired changes to code. This is sort of what I was saying earlier. No mutations are unique to radiation. So there's nothing special about radiation in the type of mutations that it causes. The type of mutations radiation causes are the same type of mutations that are already manifesting in us all the time, okay? Now, most mutations repair themselves naturally. Like if a single cell in your body has a mutation, usually that mutation's bad for the cell's survival, and the cell will either not survive, or when it makes two new daughter cells, they won't survive, okay? Occasionally, these mutations manifest in a way that is good for the cell's survival, or they manifest in a way where the cell's allowed to continue to procreate, to proliferate. And in some cases, it can be the right kind of mutation to make the cell sort of have runaway growth, and we call that cancer, okay? Mutations, though, are not unique to radiation. Radiation makes the same kind of mutations that have already been happening. Okay. Most are recessive mutations, meaning they basically are the mutations that happen to our DNA are happening in parts of the DNA that are not getting used. Okay, which means if you do hand down the mutation if this organism hands down a mutation to the offspring, if it's a recessive mutation, it's not likely to manifest. It's not likely to show up. I don't know if you know this, but I told you your DNA strand's very, very long, right? But we don't actually access all of it. It's very interesting. Our bodies only use a very tiny portion of our DNA to do what they do. A big bunch of our DNA code, you can look up how much of it is, but a big chunk of our DNA code is just extra stuff that we don't really use for anything, right? It sort of comes along for the ride. And when mutations happen they're generally not harmful. Mutations follow a linear, non-threshold curve. Like, so if you plot mutations on a graph, plot the effect from doses of radiation, it's a linear graph. More radiation exposure makes mutations more likely. Okay? As a population gets more and more radiation exposure, more and more people will manifest genetic mutations. Notice we said more and more people will manifest genetic mutations. I did not say more and more people will get worse genetic mutations. So this is referred to as an all or nothing phenomenon, okay? The percentage of people getting mutations will go up if radiation exposure to a population goes up, okay? And that's the linear part of it. More people get radiation, more people get genetic mutations. The non-threshold part of it is there's no amount of dose that we say is safe. Any amount of radiation dose can manifest a mutation. So there you go. Any amount of radiation, no matter how small, can cause a mutated gene. The incidence within a population increases proportionately to the exposure that the whole population receives. So the graph that the way this plots out on a graph is like this. A linear graph with no threshold. So genetic mutations are random. Any amount of radiation dose can cause them. More radiation dose causes more of them. It doesn't make them worse, it just causes more of them. These types of questions are going to come up a lot on our tests, state board exams, right? The dose response relationships are going to be are a big subject of radiation safety. The fact is that you have to understand the relationship between small doses of radiation and their effects, as well as large doses of radiation and their effects. The effect of small doses of radiation is to cause genetic mutation. That genetic mutation follows a linear non-threshold graph. Okay. So, we say there's chromosome damage. How do we see it, right? This thing's called a karyotype. This is a picture graph of all of the chromosomes in a single cell taken during metaphase. Metaphase is the time in the cell where all the chromosomes line themselves up along the equator of the cell, ready to be split in half. So they're already copied, which is why when you look at these chromosomes graphs, or when you see a picture of a chromosome, it always looks like an X, right? It's really two copies of the same thing. Okay, so that's why there are 23 pairs. The cell only needs 23 chromosomes. The 23 pairs are so that when the cell splits, it can send 23 chromosomes to each new cell. Okay, so if you look at chromosome number 1, right, in row A, there are two copies of that chromosome. It is exactly the same. Okay, one copy will go to one cell when the cell splits, the other copy will go to the other cell. These karyotypes basically line up so they take a picture of the cell during metaphase, a photomicrograph of the picture of the cell during metaphase, and then they, on these karyotypes, they line the chromosomes up according to size. Okay, so you're seeing largest chromosomes down to smallest chromosomes. And so it's important that we see there they are, there's some chromosomes, okay? And we see their rough shape. They are roughly X-shaped, right? Chromosomes are roughly an X shape, even though they're not a perfect X. They're two adjacent sausages. Two adjacent chromatids form a chromosome, right? So damage to chromosomes can manifest in a few different ways. Let's look at a couple of these. So photomicrographs of chromosomes after radiation exposure have been done, and these are some of them. Photomicrographs of actual visible deformities to chromosomes. So one example would be a chromatid lesion, where you have a piece of the chromosome which has broken off but stayed nearby. Lesions, breaks in the strand. Okay. Chromatid breaks, where you have physically separate pieces that are very disconnected from the chromosome. Fragments, isochromatid fragments, little pieces of chromosome just floating around. That doesn't look anything like chromosomes normally do. Other interesting things, translocations, where a piece of a chromosome breaks and attaches to a nearby chromosome. Dicentrics, where you have multiple, like if you look here, there's one set of chromosome, there's another set, they attached end to end. Okay, lots of radiation exposure, broke a lot of pieces of the strand, and those strands reattached end to end with another chromosome. Rings, where one chromosome will wrap in on itself. Or other things that are interesting too, like quadriradius, endoreduplication, triradius. Triradius here, these are quadriradius. There's endoreduplication. Lots of big names here, but the point is to show you that, chromosomes have a normal appearance. We can then expose animals to high doses of radiation and then take pictures of their cells afterward. Okay? We do this with the animals like mice and rats and things like that. And then we take pictures of their cells afterward, and we see the chromosome damage that has happened. And this is some evidence of chromosome damage afterward. Okay, so that finishes our talk about, that finishes the biology review and the talk about cell death and damage due to radiation exposure. Let's take 5 minutes here, and then I'm going to just switch gears here.